am your tutor Rajneesh Kambati. I welcome you to learn Chapter 2 of Biology Standard 11. So let's begin. Do comment on my effort how you like it or not, was it useful or not. I suggest take notebook and make notes with me while learning. History and evolution of classification of living organisms. 1. Early classification efforts. Since ancient times, humans classified living organisms instinctively. Early classification of plant and animals was not scientific, but based on utility/usefulness to humans. For food, classified into edible or poisonous plants. Shelter, e.g., timber trees. Clothing, e.g., animals with wool or hides. In ancient times, humans classified organisms not scientifically, but instinctively. They grouped living things based on their use. Like plants that were edible or poisonous, trees that could provide timber, and animals that could offer wool or hides for clothing. But remember, this wasn't science, just survival. Now, fast forward to the great Greek philosopher Aristotle, around 384 BC. He made the first scientific attempt at classifying living things, based on observable traits. He grouped plants as trees, shrubs, or herbs, and animals as those with red blood and those without it. In today's terms, vertebrates and invertebrates. It was revolutionary for its time, but far too simple for the complexity of life. Then came Carl Linnaeus, the father of modern taxonomy, in the 18th century. He introduced a two-kingdom system. Everything was either Plantae or Animalia. But this system had problems. There was no separation between simple and complex cells. No distinction between one-celled organisms and multicellular ones. And what about euglena? It behaves like both plant and animal. This system couldn't explain it all. As science progressed, biologists realized, we need to look beyond just shape or size. Classification had to include cell structure, prokaryotic or eukaryotic, type of cell wall, nutrition style, autotroph or heterotroph, mode of reproduction, evolutionary ancestry and habitat. This opened the door to more accurate systems. 5. Evolution of kingdom systems. Let's explore how the classification evolved. In 1866, Ernst Haeckel added a third kingdom, Protista, for unicellular organisms to the two kingdoms and hence becomes three-kingdom classification system. In 1956, Copeland added a fourth, Monera, group dedicated for prokaryotes like bacteria. Then in 1969, came Whittaker's 5 kingdoms, Monera, Protista, Fungi, Plantae, and Animalia. It was the most logical model at the time. Later, with molecular biology, scientists proposed even more advanced systems, like Woe's three-domain system, which included bacteria, archaea, and eukarya. Three-kingdom system of classification. Proposed by Ernst Haeckel. Year 1866. Background. Before Haeckel, living organisms were classified into just two kingdoms. Plantae. All green plants, algae, fungi, etc. Animalia. All animals, including protozoa. However, as scientific knowledge grew, it became clear that many unicellular organisms, like protozoa, algae, and bacteria, did not fit well into either Plantae or Animalia. Haeckel's major contribution. To resolve this issue, Ernst Haeckel introduced a third kingdom, Protista. This new kingdom was created to include all unicellular and primitive multicellular organisms that did not clearly belong to plants or animals. 3 kingdoms. Kingdom description examples. Plantae multicellular, autotrophic organisms with cell walls containing cellulose trees, flowers, algae, some, mosses. Animalia multicellular, heterotrophic organisms without cell walls, insects, birds, mammals, fish. Protista mostly unicellular organisms with varied cell structures and modes of nutrition, amoeba, paramecium, euglena, diatoms. Features of kingdom Protista. Mostly unicellular, some simple multicellular organisms. Eukaryotic, have a true nucleus. Some are autotrophic, e.g. Euglena. Others are heterotrophic, e.g. amoeba. Can reproduce sexually and asexually. Found in aquatic or moist environments. Limitations of the three-kingdom system. Prokaryotes, like bacteria, were still not separated into their own group. Protista included too many diverse organisms, both autotrophs and heterotrophs. No clear boundaries between simple plants, animals, and protists. Importance of Haeckel's system. Marked a transition toward more accurate classification. Recognized the need for a separate group for unicellular organisms. Laid the foundation for later classification systems like the five-kingdom system by Whittaker. Summary chart. Feature Plantae Animalia Protista. Cell type eukaryotic eukaryotic eukaryotic. Body type multicellular multicellular mostly unicellular. Nutrition autotrophic heterotrophic both. Examples: mango tree, algae lion, fish amoeba euglena. Four-kingdom system of classification. Proposed by Herbert F. Copeland. Year 1956. Why was the system changed again? The three-kingdom system by Haeckel still grouped bacteria and unicellular algae under Protista, which was too broad and confusing. With advances in microscopy and cell biology, scientists discovered that bacteria lacked a nucleus. They are prokaryotic, unlike Protista, which are eukaryotic. Hence, Copeland proposed a separate kingdom for prokaryotes. Major contribution of Copeland. He created a new kingdom called Monera to accommodate all prokaryotic organisms like bacteria and blue-green algae, cyanobacteria. The four kingdoms in Copeland's system. Kingdom description. Cell type examples. Monera unicellular prokaryotes without a true nucleus prokaryotic bacteria. Cyanobacteria. Protista mostly unicellular eukaryotes with a true nucleus eukaryotic amoeba. Euglena, diatoms. Plantae multicellular, autotrophic organisms with cell walls eukaryotic trees. Algae, ferns. Animalia multicellular, heterotrophic organisms without cell walls eukaryotic humans. Birds, insects. Key features of kingdom Monera. Prokaryotic organisms. No membrane-bound nucleus or organelles. Unicellular, sometimes colonial. Asexual reproduction, mostly binary fission. Includes both autotrophs, e.g. cyanobacteria, and heterotrophs, e.g. bacteria. Cell wall may or may not be present. Limitations of Copeland's system. Protista still included too many unrelated organisms, from unicellular algae to protozoa. Fungi were still grouped under Plantae, despite being heterotrophic and having a different cell wall composition, chitin, not cellulose. The system did not clearly reflect evolutionary relationships. Significance of Copeland's contribution. Recognized the fundamental difference between prokaryotic and eukaryotic cells. Marked a step forward in biological classification based on cellular structure. Paved the way for the five-kingdom system by Whittaker in 1969. Summary table. Feature Monera Protista Plantae Animalia. Cell type prokaryotic eukaryotic eukaryotic eukaryotic. Nucleus absent present present present. Cell wall present, peptidoglycan in bacteria. Variable cellulose absent. Body type unicellular unicellular / some multicellular multicellular multicellular. Nutrition auto / heterotroph auto / heterotroph autotroph heterotroph. Examples: bacteria, cyanobacteria amoeba, Paramecium mango tree, algae dog, human. Six-kingdom and three-domain system. Developed from molecular studies, especially rRNA analysis. Proposed by Carl Woese. Year, late 1970s. Finalized in 1990 with three-domain system. Why was the system changed again? With advances in molecular biology and genetics, scientists began comparing organisms based on DNA/RNA sequences, especially ribosomal RNA, rRNA. Carl Woese discovered that some prokaryotes, previously in Monera, were fundamentally different at the molecular level. He split Monera into two genetically distinct groups, bacteria and archaea. Evolution from 5 to 6 kingdoms. The 5-kingdom system. Monera, Protista, Fungi, Plantae, Animalia, became the 6-kingdom system by splitting Monera into Eubacteria, true bacteria, Archaebacteria, now called archaea. 6 kingdoms. Kingdom cell type cell wall nutrition examples. Eubacteria prokaryotic peptidoglycan auto/heterotrophy, coli streptococcus. Archaebacteria prokaryotic no peptidoglycan, unique lipids. Auto/heterotroph halophiles, methanogens. Protista eukaryotic variable auto/heterotroph amoeba. Euglena, algae. Fungi eukaryotic chitin heterotrophic, absorptive, yeast mushroom. Plantae eukaryotic cellulose autotrophic mango tree, grass. Animalia eukaryotic no cell wall heterotrophic dog, human bird. Carl Woese' 3-domain system, 1990. To better reflect evolutionary relationships, Woese introduced a higher level of classification called domains. 3 domains. Domain includes characteristics. Bacteria kingdom Eubacteria true bacteria. Prokaryotic. Peptidoglycan in cell wall. Archaea kingdom Archaebacteria ancient prokaryotes. No peptidoglycan. Live in extreme environments. Eukarya Protista, fungi, Plantae, Animalia all eukaryotic organisms with membrane-bound organelles. Key differences between archaea and bacteria. Feature archaea bacteria. Cell wall no peptidoglycan present. Membrane lipids ether-linked, ester-linked. RNA sequences unique standard. Habitat extreme environments diverse environments. Significance of the 3-domain and 6-kingdom system. Reflects true evolutionary history using molecular data. Recognizes archaea as a distinct ancient lineage. Organizes biodiversity more accurately and logically. Widely accepted in modern biology and phylogenetics. Summary table. Domain kingdoms included cell type example organisms. Bacteria Eubacteria prokaryotic E. coli lactobacillus. Archaea Archaebacteria prokaryotic thermophiles. Methanogens. Eukarya Protista fungi, Plantae, Animalia eukaryotic amoeba. Mushroom, mango tree human. 6. The role of plant and animal kingdoms. Through all these changes, plant and animal kingdoms stayed constant. But the organisms within them changed. Earlier, fungi, euglena, and even bacteria were grouped with plants. But now we know better. Fungi get their own kingdom. Bacteria belong to Monera. And euglena. It's Protista. All because of scientific progress. Conclusion. Classification systems have become more scientific and accurate with time. Modern classification relies on a comprehensive approach, combining morphology, genetics, cell biology, and evolutionary history. Accurate classification helps in understanding biodiversity, studying evolutionary links, scientific communication and naming organisms. 5-kingdom classification. R. H. Whittaker, 1969. Why a new system was needed. Earlier classification systems grouped organisms primarily based on superficial similarities like presence of a cell wall, habitat, morphology, external features. This led to errors. Grouping unicellular and multicellular organisms together, e.g., Chlamydomonas with Spirogyra. Grouping prokaryotes and eukaryotes together, e.g., bacteria with fungi. Grouping autotrophs and heterotrophs together, e.g., fungi with green plants. Whittaker's classification criteria. Whittaker used 5 main criteria to classify organisms. Cell structure, prokaryotic/eukaryotic. Body organization, unicellular, multicellular, colonial. Mode of nutrition, autotrophic/heterotrophic. Reproduction, asexual/sexual. Phylogenetic relationships. Evolutionary history. Kingdom cell type, nucleus, body organization, nutrition, cell wall. Examples. Monera, prokaryotic, no unicellular, auto/heterotrophic peptidoglycan, in bacteria. Bacteria, cyanobacteria. Protista, eukaryotic, yes, mostly unicellular, auto/heterotrophic, present/absent amoeba. Paramecium, euglena. Fungi, eukaryotic, yes, multicellular, except yeast. Heterotrophic, saprophytic. Chitin yeast, mushroom, rhizopus. Plantae, eukaryotic, yes, multicellular, autotrophic, cellulose, mango tree. Algae. Animalia, eukaryotic, yes, multicellular, heterotrophic, absent dog. Lion, human. Key revisions from previous systems. Earlier grouping, Whittaker's correction. Bacteria plus algae in plants, bacteria, Monera. Algae, Protista/Plantae. Fungi in Plantae, fungi, separate kingdom. Unicellular organisms scattered, all unicellular eukaryotes, Protista. Based on cell wall presence, now based on nutrition, structure, evolution. Relation to three-domain system. Domain kingdoms included. Bacteria, eubacteria, Monera. Archaea, archaebacteria, split from Monera in 6 kingdom. Eukarya, Protista, fungi, Plantae, Animalia. Conclusion. Whittaker's 5-kingdom classification was a revolutionary step. Refined the grouping of organisms based on evolutionary relationships. Provided a clear separation of unicellular and multicellular, autotrophs and heterotrophs, prokaryotes and eukaryotes. Still forms the basis of many modern classifications, though further refined by the 3-domain system and molecular phylogeny. Rationale behind 5-kingdom classification. Problems with earlier classification systems. Earlier systems grouped organisms based primarily on one visible character, presence of a cell wall, and classified many different organisms under kingdom Plantae. Included bacteria, blue-green algae, cyanobacteria, fungi, mosses, ferns, gymnosperms, angiosperms. Issues with this approach. Prokaryotes plus eukaryotes grouped together. Bacteria and cyanobacteria, prokaryotic, were classified with fungi and plants, eukaryotic. Unicellular plus multicellular organisms grouped together. Chlamydomonas, unicellular, and Spirogyra, multicellular, were both classified under algae in plants. Autotrophs plus heterotrophs grouped together. Fungi, heterotrophic, and green plants, autotrophic, were not distinguished despite major differences. Cell wall composition differences ignored. Fungi have chitin in their walls. Green plants have cellulose. Issues with this approach. Prokaryotes plus eukaryotes grouped together. Bacteria and cyanobacteria, prokaryotic, were classified with fungi and plants, eukaryotic. Unicellular plus multicellular organisms grouped together. Chlamydomonas, unicellular, and Spirogyra, multicellular, were both classified under algae in plants. Autotrophs plus heterotrophs grouped together. Fungi, heterotrophic, and green plants, autotrophic, were not distinguished despite major differences. Cell wall composition differences ignored. Fungi have chitin in their walls. Green plants have cellulose. Improvements in Whittaker's 5-kingdom system. Organism type, previous kingdom in Whittaker system. Bacteria, cyanobacteria, previous kingdom Plantae, new kingdom Monera, prokaryotic. Chlamydomonas, chlorella, previous kingdom Plantae, algae. New kingdom Protista, unicellular eukaryotes. Paramecium, amoeba, previous kingdom Animalia, new kingdom Protista. Fungi, previous kingdom Plantae, new kingdom fungi. Why were changes made? Criteria changed. From basic morphology to more scientific and biological traits. Cell type, prokaryotic/eukaryotic. Body organization, unicellular/multicellular. Mode of nutrition, autotroph/heterotroph. Cell wall composition. Evolutionary, phylogenetic, relationships. Key insight. Classification must reflect evolutionary history, not just appearance. Modern relevance. This refined system allowed clear separation of prokaryotes versus eukaryotes. Grouping organisms based on nutrition, reproduction, genetics. Paved the way for 3-domain and 6-kingdom systems. Kingdom Monera. Bacteria. The simplest yet most diverse life forms. General characteristics. Kingdom Monera consists entirely of bacteria. All members are prokaryotic, i.e., they lack a true nucleus and membrane-bound organelles. Unicellular microscopic organisms. Cell wall is present and made of peptidoglycan in most bacteria. Reproduce asexually, mainly through binary fission. Habitat and abundance. Bacteria are ubiquitous. Found everywhere. Soil, water, air. Inside the human body. Extreme environments like hot springs, deep oceans, polar snow, deserts. Some bacteria live as parasites in plants, animals, or humans. Classification by shape. Shape, name, plural, example, appearance. Round-shaped are called coccus, and plural is cocci, forms grape-like clusters or chains. Rod-shaped are bacillus, plural is bacilli, forms straight chains of rods. Comma-shaped is vibrio, and plural is vibrio, forms curved rods. Spiral-shaped is called spirillum, plural is spirilla, that forms corkscrew-like structure. Metabolic diversity. Despite their simple structure, bacteria exhibit extraordinary metabolic diversity. Autotrophic bacteria, they make their own food. 1. Photosynthetic autotrophs. They use sunlight and bacteria chlorophyll. For example, purple sulfur bacteria, green bacteria. 2. Chemosynthetic autotrophs. They obtain energy by oxidizing inorganic substances like ammonia, nitrates, or sulfur. These are important in nitrogen cycle, for example bacteria like Nitrosomonas and Nitrobacter. Heterotrophic bacteria depend on other sources. Majority of bacteria are heterotrophs. Obtain nutrition from dead organic matter called saprotrophic, or living hosts called parasites. Key points. Bacteria are the oldest and most primitive life forms. Their diverse metabolic abilities make them crucial for ecological balance. Play roles in decomposition, nitrogen fixation, fermentation, causing diseases, pathogens. Mnemonic to remember bacterial shapes. Can Bacchus visit Spain? Bacchus, Bacillus, Vibrio, Spirillum. 2.1.1 Archaebacteria. The ancient survivors. What are archaebacteria? Archaebacteria are a unique and ancient group of bacteria known for living in extreme environments where most other life forms cannot survive. Part of kingdom Monera. Prokaryotic like other bacteria. Discovered to be genetically and biochemically distinct from true bacteria. Eubacteria. Unique cell wall. Their cell wall composition is different from that of eubacteria. Lacks peptidoglycan. Contains pseudopeptidoglycan or other unique polymers. This special structure makes them resistant to high salt concentration, high temperatures, acidic environments. Habitat. Extreme environments. Archaebacteria are known as extremophiles due to their survival in harsh habitats. Type, habitat, example, role, or environment. Let's explore 3 fascinating types of archaebacteria, each adapted to survive in some of Earth's most extreme environments. First, we have halophiles. These microorganisms thrive in extremely salty environments like salt lakes and salt mines. Think of vast salt flats, where almost nothing else can survive. That's where halophiles flourish. Next, the thermoacidophiles. As the name suggests, they love heat and acidity. You'll find them in hot acidic springs, such as geysers or volcanic zones. Yellowstone National Park is a perfect example of their natural habitat. Finally, the methanogens. These microbes live in oxygen-free places like marshes, swamps, and especially in the guts of ruminant animals such as cows and buffaloes. They play a key role in producing methane gas. Yes, the same biogas used as renewable energy. So, from salt mines to hot springs to animal guts, archaebacteria have mastered survival in extreme conditions. Role of methanogens. Live in the digestive tracts of ruminants like cows and buffaloes. Help in digestion of cellulose and release methane gas. Utilized in biogas production from animal dung. Significance of archaebacteria. Ecological role. Methanogens play a part in carbon cycling and energy production. Industrial applications. Used in biogas plants and biotechnology. Evolutionary insight. Believed to be among the earliest forms of life on Earth, giving clues about life's origin. Mnemonic tip to remember types. Mighty hunters thrive. Methanogens. Halophiles. Thermoacidophiles. What are eubacteria? Let's talk about eubacteria, often called the true bacteria. These are prokaryotic organisms, meaning they lack a true nucleus. A key feature is their rigid cell wall. If they move, they do so using a whip-like structure called a flagellum. You'll find them almost everywhere. In water, soil, air, and even living inside other organisms. Cell structure. Their defining structural element is a rigid cell wall made of a unique compound called peptidoglycan. Inside, there's no membrane-bound nucleus. Their genetic material floats freely. Many also carry small, circular pieces of extra DNA called plasmids. For movement, motile types use one or more flagella. And when it's time to multiply, they primarily reproduce asexually through a simple, efficient process called binary fission, splitting in two. Types of eubacteria. Eubacteria are incredibly diverse, falling into main categories based on how they get their energy. Cyanobacteria, or blue-green algae. These are photosynthetic autotrophs. Like green plants, they use chlorophyll A to capture sunlight. They can be single-celled, live in colonies, or form filaments. Found in fresh water, the ocean, or on land, their colonies are often surrounded by a protective gelatinous sheath. You might see them forming dense blooms in polluted water. Some types, like nostoc and anabina, have specialized cells called heterocysts that can fix vital atmospheric nitrogen into a usable form. Chemosynthetic autotrophs. These bacteria get their energy not from the sun, but by oxidizing inorganic chemicals, like ammonia, nitrites, nitrates, sulfur, or iron. They play a crucial ecological role as recyclers, helping cycle essential nutrients like nitrogen, phosphorus, iron, and sulfur through ecosystems. Heterotrophic bacteria. This is the most abundant group. They get their nutrition by breaking down organic matter. Many act as vital decomposers, breaking down dead organisms. Some are incredibly beneficial to us, like lactobacillus turning milk into curd, others producing life-saving antibiotics, or species that fix nitrogen in the roots of legumes like peas and beans. However, some heterotrophs are pathogenic, causing serious diseases such as cholera, typhoid, tetanus, and citrus canker in plants. Reproduction in eubacteria. Binary fission is their main way of reproducing asexually. But when conditions get tough, many can form tough, dormant spores to survive. While not true sexual reproduction, they do have a primitive way to swap genetic material through a process called conjugation, where DNA is transferred directly from one bacterium to another. Mycoplasma. The cell wall-less bacteria. Finally, there's a fascinating exception. Mycoplasma. These bacteria completely lack a cell wall. The only ones known to do so. They hold the title of the smallest living cells discovered. They can survive without oxygen, but this uniqueness comes with a downside. Many Mycoplasma species are significant pathogens, causing diseases in both animals and plants. Key summary for revision. Cyanobacteria. Think of these as nature's solar panels. They're photosynthetic, using sunlight to make food, just like plants. Some, like nostoc and anabina, have a superpower. They can fix atmospheric nitrogen, fertilizing ecosystems. Often called blue-green algae, chemosynthetic bacteria. These are the chemical energy harvesters. Instead of sunlight, they get energy by oxidizing inorganic chemicals like ammonia or nitrites. Nitrosomonas is a prime example, playing a vital role in nutrient cycles, especially nitrogen. Heterotrophic bacteria. The recyclers and helpers. Most bacteria fall here. They decompose organic matter, but many are incredibly useful. Lactobacillus turns milk into curd, others produce life-saving antibiotics, and Rhizobium fixes nitrogen in legume roots. Though some can cause disease. Mycoplasma. The unique exception. They stand out because they completely lack a cell wall. This makes them the smallest known living cells. However, this also means many, like Mycoplasma pneumoniae, are significant pathogens in animals and plants. Each type plays a distinct and crucial role in our world. 2.2 Kingdom Protista. The unicellular eukaryotes. Definition. Kingdom Protista includes all single-celled eukaryotes. These organisms form a bridge between the prokaryotic Monera and the multicellular plants, animals, and fungi. Now let's take a look at the key characteristics of Kingdom Protista, a diverse group of eukaryotic microorganisms. Cell type. All protists are eukaryotic, meaning they have a true nucleus and membrane-bound organelles, just like higher plants and animals. Body organization. Most are unicellular, but some form colonies or even filamentous structures, making this kingdom quite versatile in form. Habitat. They are primarily aquatic organisms, found in both freshwater and marine environments. So whether it's a pond or ocean, protists are likely present. Locomotion. Many protists can move. They may use cilia, flagella, or some may even be non-motile. Their methods of movement are often linked to their type and function. Reproduction. They reproduce in multiple ways. Asexual reproduction through binary fission is common, while sexual reproduction can occur via cell fusion and zygote formation. Diversity. Perhaps the most striking feature is their diversity. Protists include plant-like forms such as algae, animal-like ones like protozoa, and even fungus-like slime molds. In summary, Kingdom Protista bridges the gap between the plant, animal, and fungal worlds, truly a kingdom of diversity. Classification of protists. The 2.2.1 Chrysophytes. Microscopic golden protists of water. Members. Diatoms. Desmids, also called as golden algae. Habitat. Found in freshwater and marine environments. Free-floating in water currents, act as plankton. Microscopic in size. Nutrition. Mostly photosynthetic autotrophs. Possess chlorophyll A and other golden brown pigments. Play a key role as primary producers in aquatic ecosystems, especially oceans. Structure and cell wall. Feature description. Cell wall made of silica, forming two halves like a soapbox with overlapping lids. Silica shells tough and indestructible even after death. Appearance often ornamented and symmetrical. Ecological importance. Major component of phytoplankton. Form the base of marine and freshwater food chains. Responsible for a large portion of global oxygen production. Diatomaceous earth. Silica-rich cell walls of dead diatoms accumulate over millions of years. This soft, gritty sediment is called diatomaceous earth. Uses. Polishing agents. Toothpaste, metal polish. Filtration of oils and syrups. Used as insulating material and mild abrasive. Key points recap. Feature chrysophytes, diatoms and desmids. Cell type eukaryotic unicellular. Nutrition photosynthetic. Habitat aquatic, both marine and freshwater. Cell wall silica-based. Forms indestructible overlapping shells. Unique trait form diatomaceous earth. Ecological role chief producers in oceans. Mnemonic to remember. Crispy shells of chrysophytes create diatomaceous earth. 2.2.2 Dinoflagellates. Bioluminescent and toxic protists of the sea. Habitat. Mostly marine, saltwater. Float freely in planktonic zones. Prefer sunlit surface waters. Nutrition and color. Mostly photosynthetic, autotrophs. Some may also be mixotrophic, combine autotrophy and heterotrophy. Color varies based on pigment type. Yellow, green, brown, blue, red. Structure. Feature description. Cell wall composed of stiff cellulose plates called theca. Flagella 2. One longitudinal and one transverse in grooves between wall plates. Cell type unicellular, eukaryotic protists. Red tides. Caused by rapid multiplication, blooms, of red dinoflagellates, e.g. goniolax. Turns the sea water red or brownish. Releases toxins harmful to marine life. Can kill fishes and other aquatic organisms. Can contaminate shellfish, harmful to humans upon consumption. Special features. Some dinoflagellates are bioluminescent. They glow at night in seawater. Show unique flagellar movement, whirling/swimming pattern. Display nuclear dimorphism, unusual nuclear structure in some. Key points recap. Feature dinoflagellates. Habitat marine. Color yellow, green, red, brown, blue. Cell wall cellulose plates, stiff. Flagella 2. Longitudinal and transverse. Notable phenomenon red tide, caused by goniolax. Danger release of toxins. Can kill fish and harm humans. Mnemonic. Dino's red spin, 2 flagella in red tides begin. 2.2.3 Euglenoids. The flexible protists. Let's explore one of the most fascinating groups in the kingdom Protista, the euglenoids. These organisms are commonly found in freshwater habitats, especially in stagnant water like ponds and ditches. Now, unlike plant cells, euglenoids do not have a rigid cell wall. Instead, they have a pellicle, a flexible protein-rich covering that allows their body to twist and turn. This gives them a flexible, almost animal-like movement. They possess 2 flagella, one long and one short, which they use for movement. You'll often see them darting around under the microscope like little torpedoes. But here's what makes euglenoids truly unique: their dual nutrition mode. In sunlight, they act like plants, photosynthesizing, thanks to pigments similar to those found in higher plants. New moon, but in the absence of light, they switch to being heterotrophs, feeding on smaller organisms. This flexible behavior is called mixotrophy. The best-known example of this group is Euglena. So remember this key point. Euglenoids behave like plants in light and animals in darkness. That's what makes them such an evolutionary puzzle. Euglena diagram explanation. Let's take a closer look at the diagram of Euglena, one of the most fascinating unicellular protists. At first glance, you can see its elongated, spindle-shaped body, which helps it move smoothly in water. Starting from the front, there's a long flagellum, a whip-like structure that Euglena uses to swim. It beats rhythmically to propel the organism forward. Right next to the flagellum, you'll notice a red spot labeled stigma or eye spot. This structure helps Euglena sense light direction, allowing it to move toward light sources, a behavior called positive phototaxis. Just below the eye spot is the photoreceptor or paraflagellar body, which works together with the eye spot to guide Euglena towards sunlight for photosynthesis. As we move inside, you can see multiple chloroplasts distributed throughout the cell. These green structures contain chlorophyll, enabling Euglena to perform photosynthesis, just like a plant. In the center of the cell, there's a prominent nucleus that controls all cellular functions. Inside the nucleus, we can also spot the nucleolus, which is involved in ribosome formation. Below that is the contractile vacuole, a star-shaped organelle that helps regulate water balance by expelling excess water, preventing the cell from bursting in freshwater environments. At the rear end, there's a region where polysaccharides produced by photosynthesis are stored. Euglena stores food in the form of paramelon, not starch like plants. Remember, Euglena is a mixotroph, It can make its own food like a plant or absorb food like an animal, depending on light availability. This dual nature and complex cell structure make Euglena an excellent example of evolutionary adaptation in Protista. 2.2.4 Slime molds. Nature's mobile recyclers. Next, we have the slime molds, the recyclers of the forest floor. These organisms are saprophytic, which means they feed on decaying organic matter, like fallen leaves and rotting wood. They're usually found in damp, shady environments. Slime molds move in an amoeboid fashion, slowly creeping over their food source and engulfing organic particles. Under favorable conditions, they merge into a massive, visible structure called a plasmodium, a large, multinucleated blob that can spread across several feet. But when conditions become harsh, like lack of moisture or food, the slime mold stops moving and transforms. It differentiates into fruiting bodies, which produce spores with tough walls. These spores are extremely resistant and can survive for years. They're dispersed by air currents, allowing the slime mold to return when conditions improve. Key point: though slime molds look and act like fungi at certain stages, their cellular structure is eukaryotic and protist-like, which is why they're classified under Protista. 2.2.5 Protozoans. All unicellular, heterotrophic protists. Some are parasitic, some are free-living. Regarded as animal-like protists. Divided into 4 groups. 1. Amoeboid protozoans. Feature description. Habitat: freshwater, marine, moist soil. Movement via pseudopodia, false feet. Shell: some marine forms have silica shells. Example: amoeba, entamoeba, parasitic. 2. Flagellated protozoans. Feature description. Habitat: free-living or parasitic. Movement: flagella. Disease: some cause sleeping sickness. Example: trypanosoma. 3. Ciliated protozoans. Feature description. Habitat: aquatic. Movement: numerous cilia. Feeding cilia push food into gullet. Example: paramecium. For sporozoans. Feature description. Structure: non-motile, form infectious spores. Disease: plasmodium causes malaria. Example: plasmodium. Summary table: protists are incredibly diverse. Here's a quick guide to major groups and their key traits. Euglenoids. Like Euglena, they're mixotrophic, meaning they can photosynthesize or absorb nutrients. Found in freshwater, they zip around using long flagella. Slime molds. The forest recyclers. These are saprophytic, feeding on dead organic matter in forest litter. They creep along using amoeboid movement. No flagella or cilia here. Amoeboids. Think amoeba. Classic heterotrophs that crawl using flowing pseudopodia, false feet. They thrive in water and soil. Flagellates. Heterotrophs propelled by one or more flagella. Some, like the parasite trypanosoma, cause of sleeping sickness, live in host blood. Ciliates. Such as paramecium. These heterotrophs are covered in tiny hair-like cilia for movement and feeding. They're primarily aquatic. Sporozoans. Entirely parasitic protists. They form spores for transmission and are non-motile in their host stage. Plasmodium, the malaria parasite, infects humans and other hosts. This amazing variety shows why protists are their own unique kingdom. 2.3. Kingdom fungi. Introduction. Fungi form a distinct kingdom of heterotrophic organisms, found in diverse habitats and show wide morphological diversity. Examples: bread mold, yeast, mushrooms, toadstools. Parasitic. Puccinia causes wheat rust. Beneficial. Penicillium, antibiotic production. Where are fungi found? Cosmopolitan. Found in air, water, soil, on animals and plants. Thrive in warm and humid conditions. Grow on moist bread, rotten fruits, leaves, e.g. mustard leaves with white fungal spots. Why refrigerate food? To slow down fungal and bacterial growth and prevent spoilage. Fungal structure. Unicellular fungi is yeast. It the only member that is unicellular. Other all members are multicellular. Yeast use for brewing and making bread in bakery products. Structurally fungi made up of thread-like hyphae. Imagine a thread alone is hyphae. This thread-like hyphae when interwoven and forms a big structure it is called as mycelium. Mycelium is the body of fungus. See diagram. This is hyphae. They are of two types, hyphae with internal division with septa called septate. Due to septa it looks like single cells with single nucleus per cell. And hyphae without it is cenocytic where it looks multinuclear condition. Cell wall is present and made up of chitin and polysaccharides. Nutrition in fungi. Mode description. Saprotrophic absorb nutrients from dead matter, e.g. bread mold. Parasites live on/in living organisms, cause diseases. Symbionts live in mutualistic association. With algae lichens. With roots of plants, mycorrhizae. Examples of uses and impact. Use/impact example. Food production yeast used in bread and beer. Disease Puccinia causes wheat rust. Medicine penicillium. Source of penicillin. Mutualism lichens, mycorrhizae. Quick summary table. Characteristic kingdom fungi. Cell type eukaryotic. Cell wall made of chitin. Mode of nutrition heterotrophic. Body structure mostly filamentous. Some unicellular. Reproduction asexual and sexual. Special associations lichens, fungi plus algae, mycorrhizae, fungi plus plant roots. Let's explore how fungi reproduce, a process as diverse as the fungi themselves. Fungal reproduction is classified into three modes: vegetative, asexual, and sexual. 1. Vegetative reproduction. This type doesn't involve spores or sex cells. Instead, the existing fungal body multiplies directly. Fragmentation. The fungal mycelium breaks into pieces. Each fragment has the potential to grow into a new fungus. Fission. Seen in unicellular fungi like yeast, where the cell splits into two daughter cells, a method similar to bacterial division. Budding. Also in yeast, a small bud forms on the parent cell, grows, and then separates to form a new individual. These methods help fungi grow rapidly in suitable conditions. 2. Asexual reproduction. In this mode, fungi produce spores without fusion of sex cells. Conidia. Non-motile spores found in fungi like penicillium. Sporangiospores. Found in Rhizopus. These are formed inside sporangia. Zoospores. Motile spores found in aquatic fungi. They use flagella to move through water. These spores are often produced inside distinct fruiting bodies, and they ensure quick spread and colonization. 3. Sexual reproduction. This mode adds genetic variety, important for evolution and adaptation. It involves fusion of gametes or hyphae from two compatible mating types, followed by nuclear events like formation of oospores, formation of ascospores. In Ascomycota, formation of basidiospores. In Basidiomycota, these sexual spores are usually formed after plasmogamy, cytoplasm fusion, karyogamy, nuclear fusion, and finally meiosis to restore the haploid state. Summary. Fungi grow and spread fast via vegetative and asexual reproduction. They evolve and adapt through sexual reproduction. Spores, whether motile or not, are the main agents of dispersal. The reproductive versatility of fungi makes them one of the most successful organisms on Earth. Stay tuned as we dive deeper into fungal classes based on these reproductive strategies. Let's understand how fungi perform sexual reproduction, which occurs in three key steps: plasmogamy, karyogamy, and meiosis. Step 1. Plasmogamy. Plasmogamy is the fusion of the cytoplasm from two compatible gametes, or hyphae. At this stage, the nuclei from each parent do not immediately fuse. Instead, in many fungi like Ascomycetes and Basidiomycetes, the result is a dikaryotic cell, meaning each cell contains two nuclei, one from each parent. This unique stage is known as the dikaryophase, and it may persist for some time before the next step occurs. Step 2. Karyogamy. Karyogamy is the fusion of the two nuclei that were brought together during plasmogamy. This forms a diploid 2N zygote, where both sets of genetic material now exist in a single nucleus. This step is brief and immediate in some fungi, while in others it is delayed, allowing the dikaryotic phase to extend. Step 3. Meiosis. Finally, the diploid zygote undergoes meiosis, or reduction division. This crucial step restores the haploid N condition, producing haploid spores. These spores are released into the environment and germinate to form new fungal mycelium, thus completing the life cycle. In short, plasmogamy brings cells together. Karyogamy unites their nuclei. Meiosis ensures genetic diversity and starts the next generation. This process is vital for evolution and adaptation in fungi, allowing them to survive in diverse and changing environments. Before we dive into the major classes of fungi, let's understand some special terms commonly used in fungal sexual reproduction. 1. Dikaryon N plus N. After plasmogamy, the fusion of cytoplasm, the nuclei from each parent may remain separate in the same cell. This cell is called a dikaryon with two haploid nuclei N plus N, but no nuclear fusion yet. This condition is especially seen in higher fungi like Ascomycetes and Basidiomycetes. 2. Dikaryophase. This refers to the life stage between plasmogamy and karyogamy, where the fungal cells remain dikaryotic. It can last for days, months, or even years depending on the fungus. 3. Fruiting bodies. These are specialized reproductive structures where the fungus produces spores, either sexual or asexual. Examples include mushrooms or sporangia. They help in spore dispersal and are key to fungal reproduction. 4. Haploid spores. After meiosis of the diploid zygote, the fungus produces haploid spores. These spores help in spreading the fungus to new areas and also ensure genetic diversity, making fungi adaptable to various environments. Now, how do we classify fungi into different groups? The classification is mainly based on three important criteria. 1. The morphology of mycelium. Is it septate or cenocytic? 2. The mode of spore formation, asexual, sexual, or both. 3. The structure of the fruiting body. What does it look like and how does it function? These characteristics form the foundation for fungal classification and help us divide fungi into four major classes: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes, which we'll explore next. Stay tuned to learn more about each class and their fascinating roles in nature and human life. 2.3.1 Phycomycetes. Habitat: aquatic environments, decaying wood in moist/damp areas. Obligate parasites on plants. Mycelium: aseptate, no cross walls. Cenocytic: multinucleated cytoplasm. Asexual reproduction: by zoospores, motile, or aplanospores, non-motile. Spores are formed endogenously, inside sporangium. Sexual reproduction: zygospore formed by fusion of gametes. Gametes may be isogamous, morphologically similar, and isogamous/oogamous, morphologically different. Examples: mucor, rhizopus, common bread mold. Albugo, parasitic fungus on mustard. Remember, phycomycetes are considered primitive fungi. 2.3.2 Ascomycetes. Sac fungi. Nature: mostly multicellular, e.g. penicillium. Some are unicellular, e.g. yeast/Saccharomyces. Nutrition: saprophytic, feed on dead matter. Decomposers: parasitic, coprophilous, grow on dung. Mycelium: branched, septate, has cross walls. Asexual reproduction: produces conidia, asexual spores, exogenously on conidiophores. Conidia germinate new mycelium. Sexual reproduction: produces ascospores inside a sac-like structure called ascus. Asci are arranged in fruiting bodies called ascocarps. Examples: Aspergillus, claviceps, Neurospora. Used in genetic and biochemical studies. Morels and truffles. Edible delicacies. Quick comparison chart. Feature Phycomycetes Ascomycetes. Mycelium aseptate, Cenocytic septate, branched. Asexual spores Zoospores/Aplanospores Conidia. Sexual spores Zygospores Ascospores in Asci. Fruiting body not prominent Ascocarp. Habitat aquatic/moist/parasites diverse. Soil, dung plants. Example Rhizopus, Mucor, Albugo yeast, Neurospora, Penicillium. 2.3.3 Basidiomycetes. Club fungi. Common names. Mushrooms. Bracket fungi. Puffballs. Habitat. Decaying wood logs stumps. Parasites in living plants e.g. rusts and smuts. Mycelium. Branch. Septate. Asexual reproduction. Usually absent. Vegetative reproduction via fragmentation. Sexual reproduction. Sex organs absent. Plasmogamy via fusion of vegetative cells of different strains. Forms a dikaryotic mycelium. This leads to the formation of basidium. Karyogamy, fusion of nuclei. Plus meiosis for basidiospores. Basidiospores are formed exogenously on basidium. Basidia are arranged in fruiting bodies called basidiocarps. Examples. Agaricus, common mushroom. Eustilago, smut fungus. Puccinia, rust fungus. 2.3.4 Deuteromycetes. Imperfect fungi. Why imperfect? Because only the asexual or vegetative phase is known. When the sexual stage is discovered, fungi are shifted to Ascomycetes or Basidiomycetes. Reproduction. Only by asexual spores called conidia. No sexual reproduction observed. Mycelium is septate and branched. Habitats and roles. Some are saprotrophic. Some are parasites. Many are decomposers of litter. Play a role in mineral cycling. Examples. Alternaria. Colletotricum. Trichoderma. Quick comparison table. Fungal classes. Feature Basidiomycetes Deuteromycetes. Reproduction sexual, Basidiospores. No asexual spores, only asexual, Conidia. Sexual organs absent, not observed. Fruiting body Basidiocarp with Basidia none. Mycelium septate, branch septate, branched. Habitat soil, logs, parasite soil, litter, parasitic. Examples Agaricus, Puccinia, Eustilago alternaria, Trichoderma, Colletotricum. 2.4 Kingdom Plantae. The green world. Key features. Kingdom Plantae includes all eukaryotic, chlorophyll-containing organisms. These organisms are mostly autotrophic, meaning they use photosynthesis to make their own food. But did you know? Not all plants are purely autotrophic. Some have developed partial heterotrophy, using other means to get nutrients. For example, bladderwort and Venus flytrap are insectivorous plants. They trap and digest insects. Cuscuta, also known as the daughter plant, is a parasitic plant. It wraps around other plants and absorbs their nutrients. Cell structure. Plants are made up of eukaryotic cells with chloroplasts, where photosynthesis occurs. A cell wall made of cellulose, providing rigidity and shape. Groups included in Kingdom Plantae. This kingdom is vast and diverse. It includes algae, aquatic simple plants, bryophytes, like mosses, pteridophytes, ferns and their relatives, gymnosperms, cone-bearing plants, angiosperms, flowering plants, the most advanced and diverse group. You'll learn more about each of these in Chapter 3. 2.5 Kingdom Animalia. The heterotrophic multicellular world. Key features. All are multicellular, eukaryotic, and heterotrophic. Cell wall is absent in all animals. Food source. Directly or indirectly depend on plants. Nutrition. Mode of nutrition is holozoic. Ingestion, digestion, absorption. Digestion occurs in an internal body cavity. Food storage. as glycogen or fat. Growth and development. Definite growth pattern. Develop into adults with fixed body shape and size. Higher animals. Exhibit advanced nervous and sensory systems. Neuromotor coordination enables complex behaviors. Most animals are motile, can move. Reproduction. Sexual reproduction by copulation of male and female, followed by embryonic development. Further details on animal phyla are in Chapter 4. Quick comparison. Plantae versus Animalia. Feature Plantae Animalia. Nutrition autotrophic, mostly. Heterotrophic, holozoic. Cell wall present, cellulose, absent. Motility generally non-motile, mostly motile. Reserve food starch, glycogen or fat. Reproduction asexual and sexual, alternation, mostly sexual. Growth indeterminate, continuous. Determinate, fixed shape/size. Nervous system absent present, in higher animals. 2.6 viruses, viroids, prion and lichens. Why not in 5-kingdom classification? These acellular or symbiotic life forms do not fit into Whittaker's 5-kingdom system. Viruses, viroids, and prion are acellular. Lichens are dual-organism symbionts. Viruses. Between life and death. Nature. Non-cellular, crystalline outside host. Obligate parasites. Need a host to reproduce. Show no metabolism outside host, inert. Structure. Made of protein coat, capsid, and nucleic acid, RNA or DNA. Never contain both RNA and DNA together. Capsid made of capsomeres, arranged in helical or polyhedral shapes. Discovery timeline. Scientist year contribution. Dmitry Ivanovsky 1892 Mosaic disease of tobacco. Filtrable agent. M.W. Bajerinck 1898 coined the term virus. Contagium vivum fluidum. W.M. Stanley 1935 crystallized virus, proving non-cellular nature. Types by genetic material. Virus type genetic material examples. Plant virus single-stranded RNA and TMV. Animal virus are with single-stranded or double-stranded RNA. Double-stranded DNA in HIV, herpes. Bacteriophage double-stranded DNA in T2, T4 viruses. Diseases caused. Humans, AIDS, HIV. Influenza, mumps, herpes. Plants. Mosaic, yellowing, curling, stunted growth. Bacteria. Attacked by bacteriophages. Viroids. Naked RNA invaders. Discovered by T.O. Diener in 1971. Caused potato spindle tuber disease. Free circular RNA molecules. No protein coat. Lower molecular weight than viruses. Prion. Infectious proteins. Nature. Protein only, no nucleic acid. Abnormally folded, can trigger misfolding in host proteins. Diseases. Disease host notes. BSE, mad cow disease. Cattle brain degeneration. CJD, Creutzfeldt-Jakob disease. Humans fatal neurological disorder. Lichens, nature's symbiotic team. What are lichens? Symbiotic association of alga, phycobiont, autotrophic, photosynthesis, fungus, mycobiont, heterotrophic, shelter, water, minerals, mutual benefits, partner role. Alga prepares food, photosynthesis. Fungus provides shelter, absorbs water and nutrients. Importance. Appear as single organism. Sensitive to pollution. Used as bioindicators. Quick revision table. Organism type, genetic material, structure, reproduction example. Virus, acellular DNA or RNA protein coat. Capsid. Host-dependent TMV. HIV, bacteriophage. Viroid, acellular RNA, only no coat, host-dependent potato spindle disease. Prion, acellular protein, only no genetic material, self-propagating BSE, CJD. Lichen, symbiotic. Dual organism, vegetative, Parmelia. Usnea.